All right, good afternoon and hello everybody. It's my great pleasure to welcome you to another ERKNet webinar. My name is Jens König. I'm a pediatric nephrologist from Münster, and I have the honor to introduce our today's expert, Professor Carsten Wagner from Zurich. Professor Wagner studied medicine at the University of Tübingen, where he also obtained his medical dissertation in physiology on phosphate transport. He later continued this way as a fellow of Professor Gerhard Giebisch and Professor John Geibel at the Yale University, focusing on epithelial transport and regulation of acid-base physiology. And after moving to Zurich, Professor Wagner worked first as a postdoc and assistant professor at the University of Physiology before receiving full professorship in 2009. His main research focus has been on various aspects of renal and intestinal phosphate transport, regulation of phosphate homeostasis, impact of phosphate on health and disease processes, as well as the genetics of phosphate balance. Currently, Professor Wagner is the chair of the Scientific Advisory Board of the European Renal Association, as well as an editor of Pflüger's Archive, the oldest journal of physiology. And today, Carsten Wagner will take us on a journey on phosphate disorders, a highly complicated and challenging topic for most of us. So thanks, Professor Wagner, for sharing your time and expertise with us.
Thank you very much for the kind introduction, and yeah, also welcome from my side to this webinar. I have a few disclosures that I'd like to share, and yeah, starting with phosphate balance and why we actually need phosphate balance. Phosphate is a critical and essential element, a mineral that is required for many different processes in our body, including the formation of membranes, cell membranes, but also intracellular membranes. It basically is the backbone of any genetic information, in DNA or RNA. It is required for signaling. Phosphorylation, dephosphorylation is one of the major signaling pathways. Of course, it is part of our energy currency, ATP. And last but not least, of course, phosphate in the form of hydroxyapatite gives stability to our bones and is therefore very essential. In fact, about 1% of our body mass is phosphate, so it's a really important mineral in our body. Now, phosphate balance is achieved basically by the dietary intake of phosphate and the regulated excretion through kidneys. And at the end of the day, these two need to match. And since the intestine, as I will show you in a minute, is rather unregulated, this is really a task that is achieved by the kidney to adapt renal excretion of phosphate to the amount of phosphate absorbed from the gut. Of course, phosphate then absorbed from the gut will be distributed in the extracellular fluid, is in exchange with soft tissue, where we have about 10 to 15% of body phosphate stored, and then of course the major storage occurs in the bone compartment, where we have about 80 to 85% of all phosphate stored. The plasma concentration or serum concentration that is clinically assessed represents in fact less than 1% of the total phosphate store in our body, and therefore this is not always giving us really an exact assessment of total body phosphate storages. Now, phosphate is absorbed from the diet. There are different forms of phosphate in our diet. Some in the form of phytates, usually in vegetables and grains. These are poorly absorbable because our body does not contain the enzymes to break down phytates. And the little phosphate that we gain from phytates comes usually from the activity of phytases in the gut microbiome. Our main sources for phosphate are organic phosphates and inorganic phosphates, and these are often added as food additives and contribute massively to the supply of phosphate in industrialized countries. Now, phosphate at low concentrations in our gut is absorbed through a series of phosphate transporters. They are partly regulated by phosphate availability and calcitriol levels, while under conditions of high phosphate supply, most of the phosphate is absorbed through the paracellular pathway, and to our best knowledge at this point, this is rather unregulated. So the more phosphate we provide to our gut, the more our gut absorbs, no matter whether we are in excess or in deficiency for phosphate. And that means that the kidney indeed needs to regulate whole body phosphate homeostasis. In the kidney, phosphate is freely filtered in the glomerulum, and then most of its reabsorption takes place here at the level of the proximal tubule, where we have a series of phosphate transporters that are depicted here on this scheme. In essence, we have three different sodium-driven phosphate transporters, NaPi2A and NaPi2C. They belong to the SLC34 gene family, and PIT2, which belongs to the SLC20 gene family. And I will come back particularly to these two transporters, NaPi2A and NaPi2C. Now, in general, in a balanced situation with a kind of balanced diet, we absorb about 80% of the filtered phosphate load, meaning that about 20% is excreted through the urine. Now, the activity of these transporters and hence the excretion of phosphate or the reabsorption of phosphate is highly regulated. And we see that these transporters are down-regulated under conditions of high phosphate intake, when parathyroid hormone is high, when fibroblast growth factor 23, another phosphaturic hormone, is high, during acidosis, when dopamine is released, or when glucocorticoids are high. On the contrary, these transporters are upregulated, so phosphate reabsorption is stimulated under conditions of low phosphate intake from diet, under growing conditions, so growth hormones or insulin are high, or in conditions of metabolic or respiratory alkalosis. So these are highly regulated and integrate a lot of signals from metabolism. Now, this is at first glance a rather complex and maybe overloaded scheme. As physiologists, we love schemes with a lot of arrows. And so what is depicted here is a triangle of three regulatory hormones that are really at the center of regulating phosphate balance. So this is FGF23, fibroblast growth factor 23, which is mostly secreted by bone. Then parathyroid hormone, obviously secreted from parathyroid glands, and calcitriol produced by kidneys. Now, when phosphate rises because we consume a phosphate-rich diet, this phosphate triggers an increase in PTH secretion and an increase in FGF23 secretion. These two synergize to induce phosphaturia in the kidney. They both downregulate renal phosphate transporters. However, there is a differential regulation also. Under conditions of highly elevated parathyroid hormone, calcitriol levels in the kidney increase, while under conditions of high FGF23 levels, calcitriol synthesis is decreased and calcitriol degradation is increased. So depending on the balance between PTH and FGF23, we may have higher or lower calcitriol levels. Of course, there are also other signals that go into calcitriol synthesis, but these two are very important, and they then influence the absorption of calcium and to a lesser extent of phosphate in the gut, and thereby closing this loop here. It's important to know also that PTH, FGF23, and calcitriol regulate each other in positive and negative feedback loops. Parathyroid hormone not only stimulates calcitriol synthesis, but it also stimulates FGF23 release. On the contrary, FGF23 high levels suppress PTH and suppress calcitriol. And calcitriol itself suppresses PTH secretion and stimulates FGF23 synthesis. So really a system of check and balance between these three hormones. However, there are, of course, then a number of other factors that go into the synthesis or the inhibition of FGF23 release, and only a few of them are mentioned here. Growth hormone, I already mentioned, it stimulates renal phosphate absorption, and at the same time it inhibits FGF23 release. And I think this makes sense because under conditions of growth, the bone needs phosphate, and so we should not waste it. And I will come back to this triangle of hormones later when we're discussing some of the diseases that affect phosphate homeostasis. Now, in the past 10 or 15 years, we learned a lot about key players of that contribute to regulating phosphate homeostasis at the whole body level. And these insights came from studying monogenic diseases that affect phosphate levels. And this is a not complete overview of some of these genes. And as you can probably appreciate, there is a cluster of genes that centers around the regulation of FGF23, and depending on the effect on FGF23, they can give rise to either situations with very high FGF23 levels or very low FGF23 levels. And then there's a second cluster of genes that centers around the kidney, and these are mostly factors that are involved in phosphate reabsorption, phosphate transporters, such as those that I just mentioned, NaPi2A and NaPi2C, and they, of course, also affect then phosphate balance. And I will go now into some of the details of these diseases here. Now, when we take a step back and think about what are common manifestations of phosphate disorders in patients affected, and I'm talking now only about monogenic diseases, not affected acquired diseases, then of course hypophosphatemic disorders, they are characterized by hypophosphatemia. We have often high calcitriol levels to compensate for the low phosphate levels. These high calcitriol levels can stimulate intestinal calcium absorption, leading to hypercalcemia and hypercalciuria. And the combination of often renal phosphate losses together with renal calcium losses predisposes patients to develop nephrolithiasis or nephrocalcinosis. In some of these disorders, we see also rickets and growth failure or growth retardation as a leading problem. Hypophosphatemic disorders, they of course are on the contrary characterized by the presence of high levels of phosphate. They have often low or normal calcitriol levels, and because of the high phosphate levels, this can lead to ectopic calcifications, and I will show you examples. Now, what is important is that there is no fixed list here of manifestations that always comes together, and I will show you that also later in some cases. So there are often variable combinations of some of these manifestations, and this is in part due to variable penetrance of the underlying gene defects and also factors that we do not fully understand. Now, I should also mention that the levels of calcium, FGF23, PTH, and calcitriol are not uniformly high or low in hypophosphatemic or hypophosphatemic disorders, but it often depends on the primary cause of the disorder, so on the gene that is affected and causes the disorder, and you will see that also as we go along. Now, I'd like to start with some of the hypophosphatemic disorders, and they are in general caused by a failure of FGF23 to increase renal phosphate excretion. So there is either a lack of FGF23 or a lack of FGF23 signaling at the level of the kidney. Now let's start first with a lack of FGF23. And to understand this type of disorder, we need to briefly discuss how FGF23 is synthesized. So we saw already that FGF23 is synthesized in bone, and which is under the control of many different factors. And the full-length FGF23 that is produced particularly by osteocytes undergoes then a number of post-translational modifications. And the two major modifications are either a glycosylation, which increases the stability of the intact FGF23, or a phosphorylation, which decreases the stability of the intact FGF23. Now, if the stability is decreased, FGF23 is cleaved by different enzymes that are not fully identified. This occurs at a defined position, and this leads to the inactivation of FGF23, basically gives rise to an N and a C terminal fragment of FGF23. If glycosylation is present, these enzymes cannot cleave FGF23, and we have hence the active and biologically active and intact FGF23. Now, mutations in this gene here in GALNT3 lead to a lack of glycosylation, increase the degradation of FGF23, and thereby lead to inappropriately low levels of FGF23, and thereby not inducing phosphate excretion in the kidney. There is also an acquired form of low FGF23 caused by autoantibodies against FGF23. Now, a second cause for a failure of FGF23 to signal can be that FGF23 needs a second protein to exert its biological activity. So in the kidney, FGF23 mostly signals through the FGF receptor 1C, which is present in the renal tubular cells. And in order for FGF23 to bind to this receptor, it needs a co-receptor, and this is alpha-klotho. Alpha-klotho is expressed in the kidney, it's present in the kidney, and basically forms a complex where the receptor, klotho, and FGF23 bind together, and only this complex then elicits the phosphaturia in the kidney. There are very rare loss of function mutations in klotho that lead to loss of FGF23 signaling. What is much more common is in acquired forms of kidney disease, where alpha-klotho is rapidly downregulated and leads then to a secondary resistance to FGF23. This is a very common mechanism in chronic kidney disease. And to illustrate such a situation of lack of FGF23, I'd like to present one patient that was seen by colleagues here in Zurich. This was a 50-year-old woman. She had been seen by rheumatology for more than 10 years for persistent joint pain. She was eventually referred to nephrology because the colleagues had noted that she had actually rather high serum phosphate levels. And on X-ray, it was seen that she had actually here quite subcutaneous calcifications in various locations. One could also see that her teeth status was not very good, and she had abnormal calcification of her teeth with canal obliterations and short roots, and she had actually undergone quite a few dental surgeries. Now, when she was seen by the colleagues in nephrology, she had serum phosphate levels of 1.85 millimoles, which is quite above the reference range. and she had also at this point already slightly reduced kidney function for her age. Now her PTH was normal, her calcitriol and calcidiol levels were normal, as well as her ionized calcium. Now, when doing a genetic test, it was noted that 3 exons of the GALNT3 gene were deleted. So this is a very large deletion in the gene. And we measured intact FGF23 in her, and this was just above the detection limit, very, very low, and particularly inappropriately low for the high levels of FGF23. So she was diagnosed with having tumoral calcinosis due to this GALNT3 gene deletion. Unfortunately, all attempts to lower her plasma phosphate levels with phosphate binders didn't fail. So she continues to be with very high phosphate levels, and she gets basically just support for her joint pain. Now, I'd like to switch now to the hypophosphatemic disorders, and I think we can group them into 2 large groups, basically for conceptual reasons. The one group is characterized by having excessively high levels of FGF23. So these are disorders that here are caused by diseases that dysregulate FGF23. And the other type of disorders is mostly here located primarily in the kidney, leading to a primary renal loss of phosphate. Now, when we look at the disorders that cause excessively high FGF23 levels, these are usually loss of functions in suppressors or repressors of FGF23 synthesis. Among them is DMP1, ENPP1, and then the most common or most frequent disorder are mutations in FEX, leading them to X-linked hypophosphatemic rickets. Now, because of the high levels of FGF23, they lead to a downregulation of the phosphate transporters in the kidney, leading to hypophosphatemia. And these patients have normal to high levels of intact FGF23. Now, even normal levels of FGF23 in face of hypophosphatemia is inappropriate. So with low phosphate levels, also FGF23 levels should be low. And anytime we find normal FGF23 levels in a hypophosphatemic patients, this already hints to a problem of FGF23. Now, when measuring 24-hour urine phosphate excretion, this can be apparently normal in these patients, but normal only in terms of amount for a patient that would be normal phosphatemic. In a patient with hypophosphatemia, we expect very low levels of phosphate. So normal levels of phosphate in the urine of a patient with hypophosphatemia is either indicative of a renal loss or that something upstream is dysregulated. It is better to measure TMP GFR. That gives a better impression of the renal capacity to reabsorb phosphate. Now, PTH and calcitriol levels in these patients can be normal to low, and as often seen in patients with X-linked hypophosphatemic rickets, there might be development of secondary hyperparathyroidism. And these patients are most often normal calcemic. Now, on the contrary, when we go to patients with primary renal losses, these are caused by loss of function mutations in different phosphate transporters or in genes that lead to a generalized Fanconi syndrome, which of course then affects the ability of the proximal tubule to reabsorb phosphate. And of course, then this leads to a reduced renal capacity to reabsorb phosphate. Again, there might be an apparently normal 24-hour urine phosphate excretion, but again, it's inappropriate when there is no hypophosphatemia, and again, it is very helpful to measure TMP GFR. Now, in this case, we have the kind of appropriate endocrine response to the hypophosphatemia, and this is normal to high calcitriol levels. This is PTH normal to low, and this is also normal to low levels of intact FGF23. Because of the often elevated calcitriol levels, the gut absorbs too much calcium, and this may lead then to hypocalciuria in these patients. So there are some commonalities between the excess FGF23 and the primary renal loss of phosphate, but there are also some distinct differences. Now, the most frequent or most common cause of excess of FGF23 is mutation of the PHEX gene. This is a gene, the function is still not entirely clear, that regulates FGF23 synthesis and secretion particularly in osteocytes, and loss of FX activity leads to a dysregulated excessive amount of FGF23 production. Of note, mutations here in DMP1 or in ENPP1 can also lead to excessive FGF23 production, and so this should be probably part of a differential diagnosis. Now, major manifestations in patients with X-linked hypophosphatemia are, of course, then driven by the hypophosphatemia and the excessive loss of phosphate through the kidneys and the suppression of calcitriol levels, and that mostly acts on bone and bone mineralization, and leads then to the typical rickets in patients, the weak bones, and then the secondary impact on joints. There are enthesopathies and very prominently also often teeth problem. And of course, they can persist into adulthood, and there are important persistences here. Now, this is just summarizing the situation in these patients. So we have this excessive FGF23 levels here in X-linked hypophosphatemia, or also in DMP1 and ENPP1 mutations, which suppresses calcitriol levels and leads to massive phosphaturia in the kidney, this is driven by the downregulation of these renal phosphate transporters and the enzymes involved in calcitriol synthesis. Now, the treatment of patients with XLH or ENPP1 and DMP1 mutations classically included supplement therapy, supplements with phosphate and calcitriol, basically to overcome this renal lack of calcitriol synthesis and to refill phosphate stores. However, in the continuous presence of this massive phosphate leak in the kidney, phosphate levels usually do not normalize but continue to increase phosphaturia. So the phosphaturia probably gets even worse with phosphate supplements. And the calcitriol supplements, of course, they help to stimulate phosphate absorption from the gut, but at the same time, they stimulate also calcium absorption from the gut, and leading to hypercalciuria in these patients. And the combination of phosphaturia and hypercalciuria can then give rise to nephrocalcinosis and kidney stones in these patients. And because phosphate levels do not really normalize with this therapy, there is an incomplete correction of mineral metabolism and an incomplete control of bone mineralization and growth. And more recently, borusumab has been introduced, neutralizing FGF23 antibodies that now in this case indeed reduce the excessive levels of FGF23, thereby allowing the kidney to produce now calcitriol, putting a brake on the massive phosphaturia, allowing also a better renal control of phosphate balance, and at least a partial restoring of phosphate balance. I think most of you may know that there was recently an update on recommendations on how to treat patients or manage patients with X-linked hypophosphatemia. And I will not go further into details here. Now I'd like to focus a little bit more on the primary renal losses of phosphate in the kidney. So we discussed previously that these 3 phosphate transporters are involved in reabsorbing phosphate from the urine and thereby help to control systemic phosphate balance. Now these transporters are part of a large protein complex that controls their stability at the apical membrane. And one important protein is the scaffold protein, which is called NERV1, which not only stabilizes these transporters in the brush border membrane, but brings them together also with the PTH receptor and signaling molecules that allow for the regulation of these transporters. So NERV1 is really an important molecule here in phosphate handling, and we know from NERV1 knockout animals that they become hypophosphatemic, hyperphosphaturic, and mimic very much a situation where these phosphate transporters are missing. Now, genetic loss of function variants in either NaPi2a, this is the SLC34A1 gene, NaPi2c, this is SLC34A3 gene, or in NERV1 have been linked with hypophosphatemia, a low TMP GFR, so which of course reflects the activity of these transporters, and the development of kidney stones and nephrocalcinosis, and in some patients also with rickets. Now we'll talk more about SLC34A1 and A3 because the situation for NERV1 is rather unclear at this point. I just want to say that all the patients that have been identified so far have only monoallelic variants, never biallelic variants, and that such variants are very, very frequent in the general population without evidence for any phosphate disorders. And so at the moment, the clinical relevance of these NERV1 mutations, I think, remains unresolved. The situation here for these other 2 genes, I think, is very clear, and I will discuss them now a bit more in detail. Now, one question that came up in the last few years was which of these transporters is actually really relevant here. And this question came up because we have knockout animals for SLC34A1 and for SLC34A3. Now, knockout animals for 34A1 have massive phosphaturia, while animals for SLC34A3 have virtually no phenotype. So in mice, this is the phosphate transporter that transports most of the renal phosphate, while this transporter is virtually dispensable. Now, we wanted to study this in a bit more detail. And what we have done is we have used a very old and classic method. We used brush border membrane vesicles. So these are basically vesicles that just contain these transporters and the brush border membrane, and where we can study the flux of phosphate into these vesicles. And we were fortunate to obtain inhibitors of NaPi2a and NaPi2a and NaPi2c. So this is a combined inhibitor. And we studied this in human kidneys from males and females around 60 years old, and in neonates in the first year of life. And we compared that to the situation in mice where we have good evidence how this works. And so by looking at the inhibition of these different transporters, we can estimate the activity of these transporters in these brush border membrane vesicles in human kidney. And I'd like just to focus here on the human situation. So this is in the first year of life. About 60% of the transport activity is mediated by NaPi2a, SLC34A1, and about 20% is mediated by NaPi2c, and another 20% which is probably reflecting PIT2. In adults, this changes. And the activity of NaPi2a contributes now less, only about 30 to 40%. NaPi2c is probably about 30%, and these PIT2 transporters also contribute between 20 and 30%. And this is totally different from mice, where in mice almost everything is continuously reabsorbed by NaPi2a. So mice are not a very good model to study renal phosphate transport, and I hate to say that as someone who has spent the last 30 years with mice studying their phosphate transport. Now, I will come back to that because I think that is important to understand why we cannot really model these diseases very well in mice. Now, what is the evidence for SLC34A1 and A3 to be really relevant for renal phosphate handling and disease? This comes first from genome-wide association studies. This is one of the first classic studies that has been done in the Icelandic population, where SLC34A1 was one of the genes that associated with the risk to develop kidney stones. And there are other genes that are well known to be involved as well. And some of these variants were actually coding variants in the transporter, and they associated in these affected individuals with low serum phosphate, low PTH. So what we would expect from a primary renal loss. Now, we have now much better evidence that SLC34A1 is really relevant in humans, and this is a study where Pete Schlingmann and Martin Conrad identified the first patients that had biallelic loss of function variants in SLC34A1. And this is one of the patients that was affected. So this is intrauterine ultrasound nephrocalcinosis. The child was born, had a failure to thrive in the first few months of life, was very hypercalcemic, had received at this time calcitriol supplements as usual, and at the same time this child or this newborn was very hypophosphatemic. And with the realization that this was a hypophosphatemic disorder, and with vitamin D was stopped and phosphate supplements were initiated, helping to restore serum phosphate levels, to normalize serum calcium levels, and to restore growth in this chart. Now, these patients are negative for CYP24A1 mutations, which is the first gene that was identified in patients with infantile idiopathic hypercalcemia, but they are either homozygous or compound heterozygous, so they are biallelic variants for SLC34A1. And we contributed here by studying many of these variants, and we could show that there are loss of function variants. So there is no transport activity anymore in these variants, and most of them are stuck in the cell. They are not making it to the membrane, so they are trafficking variants. Now, NaPi2c or SLC34A3 also causes a very similar disease, and which is characterized by hypophosphatemia. There is a clear renal phosphate leak. And what distinguishes these patients from patients with SLC34A1 mutations is that these patients often develop rickets and have low bone mineral density, while this is not the case in patients with SLC34A1. Otherwise, what is very common between patients with either SLC34A1 or A3 mutations is they have low FG23 levels, which is appropriate for the low phosphate levels. They have often normal to elevated calcitriol levels, and that drives the secondary hypercalcemia and hypercalciuria. Now, both the disease associated with SLC34A1 and the disease associated with variants in SLC34A3 were classically considered as autosomal recessive disorders, and I will also challenge this view in a minute. And this is a very recent study that looked actually at a larger cohort of families with SLC34A3 variants. And of course, the carriers that had biallelic variants, they showed a very high penetrance of disease. But the authors had also the chance to study relatives which were carriers of monoallelic variants. And these carriers showed also an intermediate biochemical phenotype. So they tended to have lower serum phosphate levels. They had a lower threshold of reabsorbing phosphate in the kidney. They had lower PTH and FGF23 levels, higher calcitriol levels. More than 30% had hypercalciuria, and there was evidence for decreased bone density in about a quarter of these relatives or monoallelic variants. And what is important is some of them were treated with phosphate supplements, but not all responded very well to this phosphate supplement. And we will see that again later in another cohort. What is very interesting is there is one very common variant in SLC34A3, and this is a mutation here in serine 192. And this variant was also found in a large cohort from a health insurance provider in the US. And among 170,000 participants in that cohort, 218 were carrying this variant as monoallelic carriers. And these carriers had a highly increased risk for kidney stones, hypophosphatemia, and lower eGFR. And this was also linked to patients that reported intake of calcium or vitamin D supplements, which of course then would aggravate the disease for the reasons discussed before. Now, what is the situation with SLC34A1? Do monoallelic variants play a role? And this discussion goes back more than 20 years, because the first report that reported variants in SLC34A1 reported 3 patients that had only monoallelic variants, but had hypercalciuria and stones. And this study was at that time heavily criticized because there was not a very clear genetic evidence for the causality here. And also the functional evidence for these variants to be functionally impaired was weak and could not be reproduced by other authors. So it was not really clear whether this were just single nucleotide polymorphisms without any clinical relevance. Now, more recently, we were involved in a study together with Friedhelm Hildebrand, studying a large number of Pakistani kindreds, where again, SLC34A1 variants were found, but all were only monoallelic variant carriers. We expressed some of these variants in oocytes, and we could show that indeed they reduced transport activity to some degree. And interestingly, when co-expressed with the wild type, there was at best a 50% reduction in transport activity, suggesting that this would give rise to a kind of haploinsufficiency in the kidney, and therefore only to a relatively mild defect in renal phosphate absorption. The problem was that we had no clinical data so that we could not really link these variants to biochemical traits, except that these patients had kidney stones of various compositions. Now, I think there was a recent study that sheds a bit more light on this, and this is a study that comes from Hanover and involved many centers from the ERKNet and ESPN. And they had collected almost 230 cases and had looked at patients that had variants in SLC34A1 and A3, and they looked at carriers with biallelic variants and monoallelic variants, but limited this to variants that were pathogenic or likely pathogenic, and I will come back to that also. Now, what is interesting here is that we can now see here what are the clinical manifestations in these patients, in these monoallelic and biallelic variant carriers. In blue, we see patients that carry variants in SLC34A1, and in black and gray, we see the patients that carry variants in SLC34A3. And what we can see is that there are some manifestations that tend to be more frequent with SLC34A1, such as vomiting and polyuria, and nephrocalcinosis, while other manifestations such as rickets and bone problems are much more common in patients with SLC34A3 variants. And this is something that had already been seen in the initial studies. However, we can also see and appreciate that monoallelic carriers have many of the manifestations, sometimes to a lesser degree, as the biallelic variant carriers. And so suggesting that indeed monoallelic variants play a role in the clinical manifestation. And I think one point that is also particularly interesting is that patients that carry biallelic variants in SLC34A3 seem to have a higher risk to develop chronic kidney disease. So this is the age-dependent decline in carriers of biallelic SLC34A3 variants, and there is a much lower level of EGFR here in a substantial number of these patients than one would expect for this age group. This is less apparent in carriers of biallelic variants in SLC34A1. This might be due to less patients in this group, and to some extent this is also surprising because SLC34A1, but not A3, has come up in various genome-wide association studies as a risk gene for chronic kidney disease. And so this is still something that certainly needs to be studied. Then there is one other point that I think is interesting, and they studied also the response of these patients to phosphate supplements. Since this is a primary loss of phosphate to the kidney, one would expect that phosphate supplements should be beneficial. And I think one point which is particularly notable is that there are differences a bit here in the response. Both patient groups responded to some extent. In the SLC34A1 carriers, there were improvements in the alkaline phosphatase and PTH levels. However, they did not reach significance, but this might be because of the relatively low number of patients in this category. In SLC34A3, this was significant, so the alkaline phosphatase levels improved, hypocalciuria and PTH levels moved towards normal levels. However, there was also a notable decline in EGFR with phosphate supplements in the SLC34A3 group, which was not seen in the SLC31A1 group. So this is something that should certainly be monitored and further investigated. Now, I mentioned that the study mostly focused here on carriers with variants that are considered as pathogenic or likely pathogenic. But there are many carriers that actually carry variants of unknown significance or variants that are considered as likely benign or benign. And this classification here that is mostly coming from in silico predictions on the effect of a mutation on protein structure and protein function, and is usually not validated by experimental data. So the question remains: what is the significance of these variants, and how should that be handled? And I will not give you a definite answer, but I'd like to show you some data that we obtained very recently. from patients that we selected from the Swiss kidney stone cohort. This is an adult cohort of patients with recurrent kidney stones. And we followed them over 3 years with deep phenotyping, biobanking, and very recently performed exome sequencing with a virtual kidney stone gene panel. And of course, we found many of the usual suspects and variants in these patients. Now, about 20% or so of the variants we found in these genes were clearly considered as pathogenic or likely pathogenic. And we had a large group of carriers, more than 50%, that carried variants of unknown significance or likely benign and benign variants. And so based just on these variants, we considered only about 8% as explained by genetics of the stone cases. What is notable is we have also a control cohort which is sex and age matched, and we found many of these variants also in our control cohort. And these are, as said, individuals where we have CT, so we know there are no renal calcifications, yet they carry many of the same variants as our stone patients. And again, we have a lot of these unknown significance or likely benign variants, which makes it very difficult to interpret them. Now, what we tried to do here is we tried to look at the biochemical traits that we would expect for these gene variants to occur if they were pathogenic, and compared now these different groups of likely pathogenic patients with likely pathogenic variants, carriers of unknown significance, and benign or likely benign, and we compared them to controls that had no of these variants and had no stones. And what we can see is for SLC34A3 variant carriers, we see that indeed they have a slightly lower TMP-GFR indicative of a renal leak. And we see that also in many of the carriers of unknown significance, suggesting that some of these variants are indeed affecting the capacity to absorb phosphate in the kidney. We see trends for higher urinary calcium excretion. We see lower FGF23 levels in carriers of unknown significance. Again, what we would expect in these patients. And we see also a higher propensity to develop calcium-containing kidney stones. So this suggests that at least some of these variants of unknown significance are pathogenic and probably need follow-up, while in the group with the likely benign and benign carriers, we do not see many biochemical alterations. And we also started to characterize some of these variants in vitro. And again, we included here this very common variant that I showed you before, where we have clinical evidence for pathogenicity. And this is clearly pathogenic, while some of the other variants that we have studied so far show very little defect. We did the same for SLC34A1 variants, and here the evidence for pathogenicity in variants of unknown significance or in the benign and likely benign group is much weaker than for SLC34A3, and so this suggests that the impact of these variants is much milder than for SLC34A3. There is one variant which is very, very common in SLC34A1, and this is a small in-frame deletion here at the very beginning of the protein. And if we look into large databases, genetic databases, we find that about 4% of the general population carry that variant with this small deletion here. The question now is this relevant at all? We studied this variant in oocytes, and in cell culture, we could not find evidence for defect. We generated a knock-in mouse, a humanized knock-in mouse. Again, this mouse has no phenotype. And we had many of these variant carriers in our group of kidney stone formers and also in our non-kidney stone former control group. And in most of the relevant biochemical parameters, we see no difference between these two groups, and we see also no difference compared to a control group. So all the evidence we have at the moment is this is a very frequent variant, but it probably is benign or likely benign and has no impact on the clinical manifestation. So with that, I'd like to stop here, and I'd like to summarize and highlight a few points. I believe inherited phosphate disorders are relatively frequent monogenic diseases. They affect multiple systems depending on the underlying gene defect, bone, kidney, endocrine organs. They can lead to ectopic calcifications. To diagnose them, it often needs a combination of hormone measurements, renal function measurement, and genetics. Of note, there is variable penetrance within a certain gene, and there is a continuum in the spectrum of disease manifestation for mono and biallelic carriers, as I hopefully have shown in the SLC34 family. We do have adults with late and mild onset, and for instance, in cohorts with kidney stones. And it can be difficult to decipher the relevance of specific variants, and we should not only rely on these in silico classifications, but it can be very helpful to do a deep biochemical phenotyping and clinical assessment of patients. And I think there are many open questions. One of them is, what are the long-term consequences of some of these variants, particularly the monoallelic variants? What are gene-environment interactions? And what does this mean also for possible therapies? And I'd like to thank many people that contributed at least to our research, not only people in my own group, but I'd like to mention particularly Ruxandra, with whom we studied this, the genetics of the Swiss kidney stone cohort. We worked a lot with Pete Schlingmann and Martin Conrad on SLC34A1 variants, as well as with Friedhelm Hildebrand and Robert Kleta, and we collaborate also with pharmaceutical company. Thank you very much for your attention.
Yes, thank you very much.
One more slide I have to show, sorry.
This is the last slide announcing the next webinar, which will just be in December, on the 9th of December, with Arend Bökenkamp presenting dense disease and a dense disease guideline. Yeah, first of all, thanks a lot, Carsten, for this very nice and well-structured overview and for sharing this very interesting new data with us. I should actually as well apologize for not telling the audience to put questions in the question box before. And so far, I actually, as a consequence, I'm afraid I don't see any questions in the question box, but you can still do that. And in the meanwhile, if I may, I might ask, I think if I got that correct, you have shown that there is differences in the expression of NaPi1A and 1C, depending on the age as well. And I was wondering, is there like differences between individuals as well, and why can't one transporter take over from the other? Or are there differences in between individuals regarding the phenotype, depending on the percentage of expression of each transporter?
I think that's a very, very difficult question. I mean, what we have is at the moment based on about 20 adult kidneys and about 20 newborn kidneys, and for which we unfortunately do not have a lot of clinical data. So we don't know how our transport assays in vitro correlates with the phosphate excretion. All we can say at the moment is that there seems to be an age-dependent relative decrease in NaPi2A activity, and NaPi2C remains or becomes equally important, or if not more important, in adults than in newborns. There might be a disease correlate. My understanding from talking to clinical colleagues is that many of the patients that carry SLC34A1 variants, NaPi2A variants, get better as they go through puberty into adulthood, while there is not so much improvement in the patients that have SLC34A3 variants. I don't know if we have the good enough clinical data on that, but our data would support that because then NaPi2A becomes maybe less important in adult kidney, and while SLC34A3 NaPi2C remains important in childhood and in adulthood. Why we are not able to compensate, this is a question I cannot answer. Apparently we need both transporters. What we know from animal studies, and I told you about the limitation from animal studies, is that at least in animals, they seem to be differentially regulated. NaPi2A seems to be a transporter that is very rapidly regulates within minutes, depending on changes in PTH and depending on phosphate intake, while NaPi2C seems to be kind of the slow guy, and maybe provide something like a background stability, and so they may serve different functions in the kidney.
Okay, thank you very much. Maybe another very basic question. I'm sorry I don't know that, but when we in the clinical practice measure FGF23, what do we actually measure? Do we measure intact or active FGF23, or do we measure cleaved FGF23 as well?
So this depends on the assay that is used. So FGF23 is not routinely measured as far as I know, and then it depends really on the assay. Now, at least in many of the earlier studies, it was mostly C-terminal FGF23 that was measured, and that was done for practical reasons, because C-terminal FGF23 is more stable and can be measured also in historic samples. If you go back to your biobank, you can measure that. Now, measuring C-terminal is basically both. It includes intact and C-terminal FGF23, so I would call it the entire FGF23. While when we measure intact FGF23, we really measure only intact FGF23. So we always expect intact FGF23 also to give lower values than C-terminal FGF23. Biologically meaningful is only intact FGF23, and measuring C-terminal FGF23 might be even misleading, because there's increasing evidence that C-terminal FGF23 can act as a basically as a counter-regulatory molecule to intact FGF23. So C-terminal FGF23 binds to the receptor, blocks the receptor, and prevents intact FGF23 to bind to the receptor. And the complex of C-terminal FGF23 but does not signal. So it would basically be a kind of decoy or a blocker. And so there would be a kind of an intrinsic antagonism here.
All right, and as I see further questions, I might take the advantage for last question. On one slide, if I've seen it correct, there was a picture where you showed the PTH receptor as well on the basolateral, but also on the apical urine side. And I was wondering, does PTH actually get there? Or what sense does it make?
So some PTH is indeed filtered, and that is true. Some of this filtered PTH can signal to the apical receptor, most of the PTH that gets into the proximal tubule is degraded there. So there are proteases such as maprin and others that rapidly degrade also PTH. So it's only in the initial part of the proximal tubule where PTH really has the chance to signal to this apical receptor. But you're right, most of the signaling occurs to the basolateral receptor.
All right, then I have a final look in the question box, but apparently you have answered all the questions with your very, very nice slides and presentation. And this just leaves me to thank you once again for this fantastic presentation. And we hope that we will see you all back again with the next webinar in latest December then. Thanks a lot, Carsten.
Thank you very much. Bye-bye.
Bye.